Okay, hi everyone. Here is our last set of video lectures, and we're going to be talking about the organ systems part 2 today. And I will have to go partially fast because I did not book the room that I'm recording this in, so I'm potentially getting kicked out soon. But okay, we're going to start with the nervous system today. And pretty much what we're going to do is learn about the neuron and kind of what all of these parts of the neuron do. So you might see a diagram like this on like your worksheet or somewhere else down the line. So it's important to know like what these parts are, what they do roughly. So yeah, let's just get into it. We're gonna start, and hopefully you can see my mouse. This top left region of the neuron, called the soma. So the soma is the main body of the cell. And this produces all of the proteins needed for all the other parts of the neuron. Also, at this end of the neuron, we have dendrites, and they're pointed to here. Hopefully, again, you can see my mouse. This is where signals from other neurons are received. So make sure you get that down. So these little branches up here are all dendrites. They're all trying to receive information from other neurons. If we travel down the neuron, we have our axon, which is this elongated fiber, and it transmits a neural signal. So, we receive a signal over here, it's traveling down the axon, and along the axon, we have myelin sheaths. And these are wrapped around the axon and help both to protect it as well as to speed up the rate of transmission of the signal. So, this is not always a really short distance, well, relatively speaking. But sometimes that signal needs to travel faster. So this is where the myelin sheath comes into play. And at the far end of our neuron, we have our axon terminal. So these contain vesicles which hold neurotransmitters. So these neurotransmitters are released and are transmitted to a different neuron, and specifically they're dendrites. So if we look back here, we could imagine that a whole another neuron is connected to our axon terminal over here. And once that axon terminal releases neurotransmitters, and our dendrites receive them, that signal will just keep going and going and being passed on from neuron to neuron. So at our axon terminal, we have the presynaptic neuron. And this is where the neurons send signals. So this is one neuron, this is another neuron. So what's happening here is we're pretty much zoomed in at our axon terminal on this side and our dendrites on this side, and we're passing those neurotransmitters in kind of the synaptic gap, so this open space, all the way to our other neuron. And there are receptors over here that are obtaining those neurotransmitters and sending the signals along. So that's kind of the basics of what a neuron is. We're going to cover a few basic types of neurons, and there's 3 types that we're going to go over. The first is a relay neuron, which is found between sensory inputs and motor responses. This is really similar in structure to the neurons that we've already seen. So these will be plugged in between neurons. And yeah, there might be something else on the far end of, whatever these are attached to, but these are the basic neurons that connect, all other neurons that are connected throughout our bodies. Now, what's not similar to this is a sensory neuron. So this is when we have a neuron that's found within our sensory systems, and they're activated by some input from the environment. So, like if it's something to do with our vision, it could be light, it could be touch, it could be any of these things. So it's receiving information at this receptor cell, and it's then passing that information on throughout the axon. The cell body is moved in this case. And then at our synaptic terminal over here. So at the end of this, we would imagine there's probably a relay neuron with dendrites ready to pass on that signal to more and more neurons. And the last type of neuron is a motor neuron. So this is one, again, a little bit different. So this nerve has dendrites, which are there to receive signals, but this nerve controls voluntary and involuntary movements of muscles and glands. So at our synapse terminal down here, we are now connected to some muscle. So we're not sending our signal to a different neuron, we're sending it to a muscle or gland to tell that muscle or gland what to do. So those are your three types. Make sure you know what these are based on the images. They're, pretty straightforward as long as you see this muscle down here, muscle, motor, this is sensory. We have some sensing receptor cell. And this is just a relay neuron. So it's passing signals from neuron to neuron. So hope this makes sense. Briefly, we're going to talk about some variation in nervous systems, because there is a lot of variation. And all animals except one group have a nervous system. And you can probably guess which group that is. The one that also lacks true tissue, that's porifera, or sponges. So these lack nervous systems. And again, these organisms have specialized cells where each cell is kind of doing its own function to help the organism survive. They're highly specialized for their function. There are a few other types of nervous systems. So nerve nets are the most simple form of a nervous system. And these have neurons spread apart, kind of just scattered throughout their body. There's no main kind of central hub that we'll see in other nervous systems. And in this case, these nerve nets in cnidarians are primarily used to sense food and to tell if they're in contact with any other substances or organisms. Next is a nerve ring, which is slightly more complex. Here there's a ring structure in the middle. We're looking at sea stars, which are in the class Echinodermata. So here they have 5 different kind of arms, right? And these all connect back at the central point. So here we have our nerve ring, and this is pretty much connecting the entire body parts or body plan of our organism. In this case, again, just looking at the sea star, the ring surrounds both the belly and mouth parts of the animal, and the radial nerves span the lengths of the arms. And also, nematodes have these. We didn't really cover what nematodes are, but they have this central nerve ring here, which spans the length of their bodies. And these are longer organisms, but they have that sort of central hub that's not super complex, but is a lot more complex than kind of these free-roaming nerves found in our cnidarian bodies. Okay, and our last type of nervous system we're going to talk about is the central nervous system, and this is what we have, as well as many other animals. Our central nervous system consists of a brain and the spinal cord. And there's also a peripheral nervous system, but the main point being is we have this sort of central hub, which is the brain and the spinal cord, where a majority of our nerves run through, and the peripheral nervous system kind of connects the ligaments of our bodies back to the central nervous system. So within our nervous system, we have both gray matter and white matter. You can see this here on this diagram. The gray is kind of purpley. So these are found in the spinal cord, as well as the brain, and they're responsible for many reflex actions and communications with the brain and the spinal nerves. Typically, gray matter holds most of our nerve cells, so kind of centralized. Um, White matter, on the other hand, contains nerve fibers which connect different chunks of gray matter areas. So, again, all connected. This is kind of found throughout our central nervous system. And our central nervous system controls a lot of different functions, including thoughts, memories, emotion, touch, vision, motor skills, breathing, temperature, hunger, probably the boredom that you're experiencing right now. Pretty much anything that you can think of is, partially controlled by the central nervous system. So it's the hub. Again, we're kind of in the organ system sections where we could talk about, these individual systems for an entire class. This is me teaching it, so it's going to be a little bit more chaotic and kind of rough over the edge. So with mammals, we typically think of these as having larger brain to body ratios than many other animals that have a central nervous system. But there are many other groups of animals that do have a central nervous system. Within our brain specifically, we see a lot of increased folding, which helps, if there's more folding in brains, like, this dolphin brain, it's really wrinkly. That helps increase the surface area of the brain, and pretty much, is a way to have a more surface area with the same volume of space. And typically we think of the surface area correlating with complex behaviors within animals. So, we can see our brain here compared to that of a chimpanzee or a cat. Moose definitely has a bigger brain than this, but that's not the point. So other animals that have central nervous systems are like planarians, which are part of the group Platyhelminths, as well as arthropods. They all have kind of these nerves that run through their bodies and kind of come back to a central point. These are just less complex than what we have, though, and what other mammals have. And I think this is cool, so you're going to listen to it. Whether or not you think it's cool or not is different. But it's really difficult to predict the evolution of nervous systems, as these are really, really rare within the fossil record. So kind of determining when these sort of specialized things popped up is really difficult to do. And the predicted order of how these evolved is nerve net, then nerve ring, then central nervous system. And I think this is just due to complexity. But I found a paper when I was Googling around that actually finds a fossil. And this image is taken of the fossil under normal lighting conditions. This one is taken using polarized light. And then this one is taken with polarized light and with the fossil being underwater. And, this is really cool because it highlights the nervous system of this creature, whatever it is. And this is approximated to be about 500 million years old. So this is, just an inkling that central nervous systems have been around for that long, more than likely. And also, I thought this was fun, but this is what the researchers illustrated the animal as looking as. So yeah, not like anything that we've seen today, but you could imagine what it would look like. Okay, so relating to nervous systems, we're going to talk a little bit about sensory systems. The five senses, you've all heard about these before: eyes, nose, hearing, touch, and taste. And we're just going to go through pretty much each of these, how these function, and how some of these might be kind of useful for animal form and function, specifically with like finding mates, because that's something that's done with a lot of these senses. Food's also important, but you find a lot of cool examples with mate selection. We're going to start with vision. So here's a little diagram of our eyes, and if we zoom in to the back of the retina, we can see kind of what's back there. So light travels through our cornea and the pupil and the lens here, and then scatters to the back of the retina, as seen here. And you can see here that we have cones as well as rods in the back of our retina. Rods specifically help us see in low light conditions. Cones, on the other hand, sorry, that's on the next slide. These rods have no color information at all, and are kind of feature very low spatial acuity. So if we were just visualizing with our rods, everything would be kind of blurry in comparison to how we normally see. And there's roughly 91 million rods on the human retina. Cones, on the other hand, allow us to see color. And these are specialized for specific types of wavelengths of light. So most help us see blue, red, and green light. So there's individual cones for each of these. These have higher spatial acuity, so they help us, see more clearly. But there's only about 4.5 million of these on the retina compared to 91 million. So it's a big difference. But the reason there are so few is because we use these in times when we see a lot of light, whereas these are used in dark environments. So there's a trade-off. You don't really need as many when you're only using them to see color, because that just won't come through when there's not enough light in the environment. Not all color vision is equal. So a lot of animals are able to see UV reflectance, and you can see some reflectance here in these like white spots on these butterfly wings. And butterflies are just one example of animals that feature a different sort of visual system as we do. They have UV cones in place. I don't think they have red cones. So they see blue, green, and UV. Bees also are a little bit different. They don't see red. And some animals have just evolved to see better in really low light conditions. So, we've talked about these eyes before. But there's so many of these because it's really hard for these guys to see where they're located, these scallops. So the more eyes, the more light you're going to gather, a bit more information can be pulled away from that then. And we've also talked about this example before, where we see disassortative mating, where tan stripe and white stripe sparrows are mating with the opposite color. Males of the white stripe mate with the females of the tan stripe, whereas males of the tan stripe mate with females of the white stripe. And my extra credit for this exam is going to be watching this Life in Color series, narrated by David Attenborough. There's 3 parts. Tell me 3 things that you learn about from each part with a timestamp, and I'll give you extra credit on this next exam. Vision School. There's a lot of cool stuff out there. You can do a lot with really specialized cameras. So it's really it's a fun documentary to watch. Hearing. Okay, so this is a bit more complex. We're gonna just kind of scan through this. Here's a little diagram of how sound moves throughout our ears. So it travels it enters through the outer ear, and these sound waves hit our eardrum and essentially make our eardrum vibrate. And these vibrations send more vibrations to these tiny bones in the middle of the ear. Sorry for the typo. So our middle ear is composed of 3 small bones. So it's highlighted here in the middle of this image. We have the eardrum, and that vibrates to send vibrations to the malleus, the incus, and the stapes. So these are all used to help amplify the vibrations within our ear. We're in the middle of our ear now. And this sends these vibrations to the cochlea. And that's this kind of snail looking thing over here. So the cochlea is filled with fluid, here's a diagram of it on top. It's on this right side here. So pink is the middle ear. This blue highlighting is the inner ear. So these vibrations cause waves to be sent along the basilar membrane, which, if you look closely here, it's located inside the cochlea. Here's what a wave looks like traveling through it. And these waves help trigger or cause small motions in hair cells found within the basilar membrane. So this, you can see the hair cells here. When these move up and down, though, they send electric signals to the auditory nerve, which is connected to the inner ear here, connected to the cochlea, and this directly sends those electric impulses to the brain. So we're going from, sound waves entering the ear, more vibrations in the eardrum being sent to the tiny ear bones, sending those vibrations to the cochlea, and here is where your electric signals are sent to the auditory nerves. So, a lot of passing on of waves. And if we, look historically at what people have done with kind of mate selection, a lot of research has been done within Darwin's finches. So, we're looking at sound waves, shocker. And we're looking at the timing of waves here. So there's different pitches, which are determined by the frequency, as well as different timings of these frequencies. So longer and lower pitches are often used for different sort of calls, either territorial calls or, birds looking for a mate. So these are used, tremendously by us as well as many other animals. Auditory cues are really important, and many behaviors stem from these. Okay, so touch is slightly more complex. Don't memorize all of these sort of parts. Just know that different sort of receptors in our skin are responsible for detecting things like light and heavy pressure, as well as hot or cold senses, and things like pain. So we have pressure sensitive receptors as well as fine touch receptors. So, if you want to find something or feel a very fine shaped object, you're going to be using these receptors. And then we have free nerve endings, which detect temperature and pain. So again, don't memorize, not on the test. Just looking at pressure receptors, we have Ruffini's endings. So these are skin stretching, movement of finger positions. We have Krauss's end bulbs. So these sense light and heavy touch, vibration, and temperature. And then we have Pacinian corpses. I butchered that. It's okay. These sense vibrations as well as deep pressure. Then if we look at our fine touch receptors, we have Messinger's corpuscles. Yeah, that's okay. Light touch, small vibration. It's here on the left. We have Merkel discs, which sense pressure, position, and allow us to feel fine edges and shapes. And then we have root hair plexus, which is super sensitive to incredibly light pressures. One thing that I want to make note of with touch is that receptive fields often vary in size depending on location of the body. So small receptive fields allow for high resolution, but large receptive fields, seen down here, don't allow for high resolution. It's kind of like you're only getting a glimpse of what's happening. So oftentimes, and I've heard about some people doing this, if you have someone poke you on the back in one or two places that are kind of close together, it's really hard to tell if there are multiple things poking you, just because we have really large receptor fields on our back. Whereas other ligaments of ours, we have smaller receptive fields. They're detecting very easily that there's one or two things. So these boxes here are pretty much receptor field sizes. So you can imagine on your hands, or somewhere more sensitive, we have, 3 in the same area as this one receptor field on our back. So on our back here, if we had two senses, two stimuli on the same block, we would only detect it as one because we're only detecting this whole area as one block. So kind of cool. My random fun fact for this spot. I combine smell and taste, and that's because both of these use olfactory receptors, at least to some degree. And these send odor signals to our brain. And here is kind of a close-up of these receptors. These typically are inherited traits. So these aren't going to be identical by people, but many people do smell things differently. And one kind of gross example of this is there was a sweaty t-shirt study done at a college. So men carry different genotypes at this MHC gene. And they had these individuals wear t-shirts essentially for 48 straight hours. And then they had females, they had 58 smell these different shirts from all these different males, and they preferred the scent of men that had opposite genotypes at this one gene. So kind of gross, but again, used in sort of a selective sense here, just for scent. And there's many more ways that scent are used for like mate selection and things like that, but this is just one really gross example, so I thought I'd show you. We detect flavor using both our olfactory and taste bud information. So here's what a taste bud nerve looks like. Flavor is learned and inherited, and we can build up a tolerance to foods that we typically or didn't like initially. We're not going to go into the fine parts here. There's different sorts of taste bud nerves that sense different sort of flavors. But all you need to know is we use both taste buds and our olfactory nerves for tasting information. And one example of this is there are super tasters. And there's a gene for this that kind of determines if you're a super taster or not. It's a dominant gene. If you have a copy, you can taste this PTC, which they put on a paper to determine if you're able to taste it or not. And this genotype pretty much means that you can tolerate or can't tolerate certain foods. Like if broccoli is super bitter to you, you're probably a super taster. You can't use that as an excuse for everything. But there are a list of foods that super tasters historically just cannot eat because it's really overwhelming to their taste buds. Okay, switching gears slightly again. We're going to talk about circulatory and nervous systems. I'm going to go kind of quick because I've been rambling. So we're going to talk about what is the nerve or circulatory system. And this just circulates blood and lymph throughout our bodies. And it consists of a heart, the whole system consists of our hearts, blood vessels, lymph vessels, and glands. And there are two types of circulatory systems. There are open circulatory systems and closed circulatory systems. So within an open circulatory system, these lack veins and arteries to deliver blood to each other. So they're kind of just releasing blood into the entire body cavity of these animals. And this is really common with arthropods or mollusks. Closed circulatory systems, on the other hand, have veins and arteries that they're helping move blood through these specific paths to distribute blood throughout the rest of the body. So us, we're a solid example of having a closed circulatory system, as well as annelids, fish, reptiles, and mammals. So these are pretty widespread and a bit more complex. So this is more simple. You're having a heart pump blood into a body cavity versus individual veins and arteries kind of sending these fluids throughout the body. And again, there are some that don't have circulatory systems. Porifera aren't alone on this one, as cnidarians also lack circulatory systems altogether. So we're just going to glance over the parts of the nerve or circulatory system. I should have filmed these as two things. But each part we're going to cover briefly. We're going to start with blood. So this helps maintain specific pH, temperature, and osmotic pressure throughout our body. And it's really important for kind of homeostasis, as well as transporting things like nutrients, hormones, and gases throughout our bodies. So blood is made up of plasma. So there's a liquid portion that includes water, protein, salts, and lipids. And then there are cells within our blood, including red blood cells, which help transport gases, as well as white blood cells. And these help us fight infections. And there's also platelets. And these are small cell fragments that help clot sort of cuts that we have in our bodies, pretty much in order to stop bleeding. So if you ever have a cut and it's like, just a little hairline cut, it's going to fill up with these platelets really quick, and hopefully you won't be bleeding for too long. So digging a little deeper into just red blood cells, these contain hemoglobin. And hemoglobin contain iron, which oxygen is able to bind to. And this iron gives red blood cells its color. So there are a few diseases, like sickle cell disease, where we see this sickle-shaped red blood cell. And this blocks a lot of blood vessels and kind of clogs things up. Okay, so this is really common where malaria is present, and that's because malaria is not actually able to grow well using these sickle cell shapes. So it's sort of an advantage to have these, odd shaped cells and just hope that your arteries aren't going to block up, just to avoid that disease altogether. Okay, so talking a little bit about cellular respiration, just the process of converting glucose and oxygen to essentially energy. And this is different from the respiratory system, but still really important. So our cells need sugar, oxygen, in order to produce energy as well as CO2 and water. And these are byproducts of cellular respiration. So oxygen is important for obtaining energy needed for just regular cell functions. And that's the case with almost all animals, Oxygen is really just key. Okay, so our respiratory system is responsible for obtaining that oxygen that's needed for cellular respiration and for creating energy. And we're obtaining that using our lungs. And this is where our red blood cells obtain oxygen and distribute it through the rest of our bodies. So oxygen gets into our lungs when we breathe. Air goes through our nasal canal, through the pharynx, through the trachea, into the bronchioles found in our lungs. And during this process, air is warmed, humidified, and filtered using our cilia and mucus to help remove any particles. And the diameter of our, this opening throughout our body gets smaller and smaller as we travel down towards the lungs. Once we get to the lungs, we see our alveoli, which are, and these are made of really thin cell walls. And these come in direct contact with our capillaries. That's here, this little thing below the alveoli. And these are found at the terminal end of our bronchioles. So this is where gas exchange occurs within vertebrates. So blood comes in, holding carbon dioxide, and it's sending that carbon dioxide into the alveoli. And while it's passing this kind of barrier where they're touching, oxygen is then being added into our red blood cells to be passed along through the rest of our bodies. And this diffusion of gases is passive, so it's just driven by this difference in concentration of this is a high oxygen area, this is a high CO2 area at one point. So that high oxygen level from the alveoli passes into the bloodstream, and the high amounts of carbon dioxide are passed into the alveoli and are then passed out of our bodies. And gas exchange is governed by Fick's law of diffusion. You don't need to memorize this, not super important. But essentially, we have things like this is the rate of diffusion. This is the area over which diffusion takes place, A. This is the pressure difference between the two sides, P. And this is the distance over which diffusion occurs. So pretty much we have evolved to be really optimized for this law of diffusion to occur because we have really high surface area in our alveoli. So you can see they're kind of like grape-like shapes. You didn't see that from the previous image. But this really leads to a high surface area in these areas. It's also really thin-walled in terms of like how many cells are actually contributing to this structure. So it's really thin, decreasing the distance between these and the bloodstream. And the concentration difference that we already mentioned back here. So it's a really high concentration difference, meaning that this is just able to occur at a rather efficient rate in combination with all of these other things. So we're really optimized to have, gas exchange occur in these organs, which shouldn't be a surprise, but it's just kind of a cool thing that we see from an evolutionary perspective. So my last couple slides, there's a few other ways that animals have evolved respiratory systems. For example, gas exchange in earth and flatworms happens actually across their skin. So they're just, they're containing CO2, but they can just kind of exchange the gas through their pores and obtain oxygen from the environment. Another example is fish. So these use gills, which are areas where gas diffusion occurs. And my last example, insects use what we call spiracles. And these are like, again, just little pore-like structures where they're able to exchange gas with their internal CO2 levels and with oxygen in the environment. So I hope this wasn't too long. Thank you guys for listening.